Okay, how's everybody doing? Ready to get going? All right. So, it's nice to see everybody again. Did you have a nice weekend? All right. So I think when we finished off last time, we had been talking about some basic auditory pathways, and in particular, let's see here. We had been talking about some basic auditory pathway processing, and we left off, and I said that you guys should come prepared to talk about some interaural timing differences. Do you remember that? So if we stick with that model that we talked about last class in terms of trying to see if you're ready to be tested today. How many of you guys feel like you would do okay if you were tested on interaural timing differences? That's okay. Why not? Becomes the question, right? So this is the chance for the next 2 hours for you to tune up anything that you feel might not be ready for being tested. Right? Cool, so let's do that. What would you not score 100% on if I asked you to describe, let's say, interaural timing differences in the auditory system? No, I do not mean the tonotopic organization, as it turns out, but that's a really good question, all right? Okay, so we'll get there, and we'll take a look at it as we move along. So, just to review, where we had been was, we had been looking at sound traveling from the outer ear to the middle ear, through the inner ear, into the brainstem, to the inferior colliculus, to the thalamus, that is the medial geniculate nucleus of the thalamus, up to the cortex, particularly the superior temporal gyrus, called the auditory cortex. There was a tonotopic organization there in the auditory cortex, just like there was in the inferior colliculus, just like there was in the basilar membrane of the cochlea. You guys remember all that good jazz? All right, cool. So that's kind of a little bit where we left off with, and we talked a little bit more about how that tonotopic organization is kind of preserved. So this is something that we looked at, which is a slice through the midbrain, or that is the inferior colliculus of, in this case, it looks like a rodent or a monkey or something like that, but it's a lower species animal. And we're looking at that inferior colliculus that we see those different colors in. Can anybody tell me things about those colors? Thanks. What do you guys remember from the book?
We relate to the frequencies you hear in the cochlea.
Yeah, so that's what those numbers are. This relates to certain areas of frequency processing, absolutely. Yeah. Anything else? Stick out? It's kind of a hint about it sticking out. What did the book say about it? It's like a paragraph about this. So, it turns out, do you guys remember, do you guys remember what it said this figure was obtained from? What kind of method? So if you stain the inferior colliculus with, let's say, 2-deoxyglucose, 2-GG. You guys remember reading about that at all? All right, so do you remember what it said? Maybe not.
When you say method, do you mean like, it said post-mortem? I don't know if that's what you mean.
Yeah, so when you stain for using 2-deoxy, 2-GG, it shows where neurons were active, which is how you can get a map like this. So it talks about that in our chapter, and one of the things that it talks about is the idea that it's not just that a particular area might be sensitive to, let's see, 15 kilohertz of sound activity, but that when this is active, what does it do to its neighbors? It inhibits them, yeah. What's your name?
Jasmine?
Jasmine, yes, great job. Okay. Yeah, so what you get is some sort of, you can think of it sort of an amplification process. So you have different sounds coming in, and as you have this tonotopic frequency organization, it actually amplifies that information, that signal as it's moving up through the brain. Right? Because we know that there's a lot of random stochastic activity happening too, and so it's more helpful to differentiate the signal that's coming in, if not only if you can activate that signal, but if you can quiet down the neighbors around it. Isn't that going to make it stand up a little taller? You guys remember any of this? Ringing a bell?
There was a figure about it you had showed us.
Yeah, yeah. This is the figure, I think.
I think it was more related to our division one.
Oh well, that's the visual system. Yeah, but we can. You're absolutely right. We can start to draw these connections now between these different sensory systems, and seeing that there's sort of common properties that seem to be evident and shared across modalities, right? In terms of general organizing principles of the brain. Pretty cool, huh? Okay. Okay, so we talked about that a little bit, and that signal gets processed up through the cortex, and we can see these different areas of cortex that are active just when you hear stuff. Let's say that's superior temporal gyrus, but then when you actually hear noises that are sounds, that have meaning, that have representation, it tends to recruit larger areas, more diffuse areas in those auditory cortex, as well as supporting areas in the parietal cortex too. So what this helps us to understand is the idea that we have basic auditory processing, but these areas are also active during times when we have richer auditory information coming in, but particularly when there's meaning for it. So who remembers reading about people who are, I don't know, let's say hard of hearing, some of these figures? You guys remember what it said about that? Yeah.
I just had a question, but
Yeah.
it's related to this. So then, okay, so I guess what it said in the book is that people who are able to lip read instead of it being, so if they're deaf, I don't know if it was if they're deaf or just hard of hearing, but they're able to still have like those parts of the brain and this recognize speech fire even though they can't necessarily hear it.
Yeah.
I'm confused on that too. So, kind of similar, so like if you're hearing like a different language, so if it doesn't have meaning to you, like if you don't know that language, that would be in the noise category, but then
Blah, 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 blah.
yeah, so if it's
Yeah,
not
that would be
language,
noise. Yeah, so if you don't know language, right? So how many of you guys speak Spanish or another language? Okay. So when I grew up, I didn't know Spanish. So I would just hear like the La Bamba music. You know that song? Para bella La Bamba. Right? And all I could hear is, I remember as like a 5-year-old, it just sounded like, and I remember because I thought I was really cool, like singing the lyrics to it like my grandma. Now it's like, ba 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 bamba. Because that's all I heard. Right? So just noise. It just sounds like random noise if you don't know that language. Yeah. And I'm sorry, you were saying another part too.
Well, no, I thought it was a different question. So
Okay.
then if people are, I just don't know if it said specifically if it was for people that are deaf or just hard of hearing, but if you're born deaf, could they potentially still have the speech recognition, like stuff firing in their brain even if they've never experienced speech?
Yeah, potentially can. Particularly, that's kind of the idea that all this is getting at, right? Which is that, yeah, you have basic auditory processing here in sort of this area, so you might call it noise, but as you get to more language and speech sounds, so language is a conceptual representation, right? That's the difference between language and noise. That's why I just said that La Bamba example, right? Like, if you just go ba 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 ba, that's not really language, that just sounds like mush. So that's noise, but it doesn't have conceptual meaning, conceptual representation that means something to us, right? You can't communicate a code to someone. And so this shows us that there's these broader networks, these other areas of the brain, very closely related to our auditory cortex, and in some cases overlapping with it, that is involved in processing that language, of course, but that we know that not all language is verbal, or that you can have sign language or body language or whatnot, and people who might be congenitally deaf, that means they're born deaf, and you use sign language instead, or lip reading, you can see the similar areas active. And do you have a question, Sean?
Kind of. So
Yeah.
your brain, like if they've done this with language, it kind of shows up more as noise, or does the brain recognize that communication is happening even if you can't decipher?
Well, it's a good question, right? Because you have to make that transition at some point, right? Developmentally. I don't know the answer on the precision level of that question, but this has already crossed the bounds into recognizable language. When they talk about speech, that means we can tell that it has meaning. Noise can, for our purposes, and the way I refer to it, noise can be, let's say, language, somebody else's language, you have no idea what it is. So like, if somebody speaks Chinese, how many of you guys would understand that? So that would just sound like just about every random stereotype of random Chinese sounds that you can make, right? It would just sound like that. We would consider that noise, right? Kind of like my La Bamba example when I was a dumb 5-year-old. So yeah. Yeah.
Similar to the blind sight, right? Even though they can't actually see it, like they're still able to describe the item. Similar?
I don't know if it's similar to blind sight, unless you can help me understand the concept that you're referring to that is related. I might just not be understanding what's being asked, and that's okay.
Yeah, I can't remember. I just remember reading something where it was talking about blind sight, about how even if the person, obviously they're blind so they can't see it, but they were still able to identify an object, even though they couldn't see it. I don't know how to explain.
Yeah, no, that's totally right about vision, but I don't know I'm not picking up on the connection with the auditory system. It might be there, I just don't know what you're asking. That's cool. If you can think of a different way to ask it, then hit me again, and we'll see if we can do it. Okay, so we talked about that a little bit, right? Because we're moving up to that. Yes, ma'am.
So you said people that are born deaf, like when they see other people signing to them, these same regions light up. So then is it really a region that is associated with speech sounds, or is it just a region that's associated with like focusing or with understanding and comprehending things? Like right now, since I'm talking to you, those regions would light up, but if I was speaking about something that just didn't interest you at all and you like zoned out, would these same regions light up?
This is a very good question. They would light up if I heard you and I knew the language. Doesn't matter if I'm interested in it or not. But if I didn't know the language and I just heard like mush garbled, like if I'm sitting up here and I'm speaking Russian or something, I'm like, я изучаю русский язык, русский язык 3 года, right? You could hear that for a moment or two and then you're going to start zoning out. And it's not, it just sounds like gibberish, right? Yeah. So anyways, so we get this idea of that auditory processing moving up to the cortex there, and we can start to sort of see how this information can be encoded then, so that it's how it is that it can then be deciphered. And there's two big ways that people talk about it. They talk about discriminating frequencies in terms of place coding and temporal coding. So anybody remember anything about place coding and temporal coding? It's a good thing I didn't throw a quiz out there today. Alright, so when we have place coding, that's referring to basically information being encoded in the place of where it's detected. So remember we had those different tuning curves in the basilar membrane and the cochlea? Different spots along it. Somewhere along there is Megan Trainor's house. Right? It's Megan Trainor's house. Why? No treble. Yeah. I used to throw a little like GIF in there of her like dancing or something. But yeah, right, so there's spots on there that care very particularly about specific frequencies and don't care about other specific frequencies. And so that's about the place on the basilar membrane, the place in the cochlea. So remember that basilar membrane kind of fits in the cochlea, So I figured out this analogy in this example last year. How many of you guys ever eaten crabs? Delicious, right? You guys ever go to like Crab Shack or something like that? Get special at the grocery store. So then, how many of you guys haven't eaten crabs before? A couple of you guys? Okay. Highly recommend it. It's delicious. Totally should. Now, let's say you're not a seafood fan or something, so you're not going to eat it. When you have the crab claw, you guys who've eaten crabs before, you know that the meat, do you guys know how the meat, right, you have to crack the crab shell claw, or the crab claw shell, and inside there's the meat. And you know how around it, or I should say the meat is around this middle kind of thin bony membrane? You know that middle sort of sheath in between? It's almost like a fingernail for the crab. Do you guys know what I'm talking about? Right? You definitely don't eat it because it's like a part of like the structure, but you pull the meat out from around it. It's that thin little. Maybe I'll bring crabs in one day or something. We'll eat some crabs. It'll be delicious. That's a really good idea. We'll get some crabs. Yeah. That's fine. You don't have to eat them. I'll eat them. More for us. So anyways, when you do that, you can see that there's that membrane. There's that physical structure, and it's inside the meat on the crab claw. That's what the basilar membrane is like through the cochlea. So when we say there's place coating, and I say somewhere along the basilar membrane or along the cochlea, it's kind of referring to the same thing because that basilar membrane is slid in the middle of the cochlea. You guys on board? Okay. So what's that saying is that the pitch, right, particular frequency sort of sound, can be encoded based on where that receptor is coming from, sending those trains of action potentials being depressed by the basilar membrane, raising those hair cells up against the tectorial membrane. You guys remember we talked a lot about that? Okay. So, in that regard, if an action potential is being sent from somewhere by the base of the cochlea, that's going to signal the treble. And if it's near the apex, remember the apex? So you roll that cochlea out. If you roll it in, roll it out. If you roll it out, the apex is like your fingertips. Right? The apex of a pyramid is kind of like that point on top. So this is like this, and it rolls out. So if you take it this way and you roll it up, that's the apex up here. So now you have the apex rolled out this way. And the apex is floppier. Like those guitar strings we talked about for detecting bass. Right? Which is a little confusing because you have two different kinds of bass here, right? You have this bass and this bass. So be very careful. If you try to just rote, regurgitate, memorize this stuff, you're going to lose the game. You're just going to lose. Sorry. It's just not how the game's rules go in science. Just memorization doesn't matter. We'll have robots doing those jobs soon. We don't need you to memorize stuff. We need you to think. The robots can't do that. That means you got to study and remember this stuff in ways that make sense, that connect it together and use it and practice it rather than just remember it. Because if you just try to remember stuff, you're just going to remembering bass and bass. And you're going to be confused, even if you're the ace of bass. Yeah, some of you people old enough know what I'm talking about. That was an old band in the 90s. But they had a good song or something. You know, it's like, I saw the sign. I saw the sign. I saw the sign. Yeah, that was ace of base. So, base here, B-A-S-E, means the bottom. That is the base of the cochlea that's going to connect essentially to that oval window. Base here means frequency. Low sound, the bass of the band. Right? As opposed to treble, which is right? High frequency. So anyways, so that's the idea with place coding, is that different spots in there is how you can determine what the sounds are that's happening in the outside world, giving you information of what the heck is happening out there. The other idea is that the information can be coded in terms of timing, that is temporal coding. Okay? So that's about the timing. So the rate of firing of these neurons. So when they fire, how fast they fire, can encode how fast or the frequency of that auditory stimulus. So for a period, or a window of these kilohertz sound waves, there's going to be a relative one-to-one mapping. So that is, the brain cells themselves the hair cells themselves will fire at that rate of that wave. Now there's going to be a point at which it can't keep up because it gets way too fast. And then it'll start to fire essentially on a multiple of that rate. Sort of a derivative, which is pretty cool. The brain's able to figure that out. So the idea is that they both kind of work together so that we can understand a little bit about what's happening in that auditory information. So it's place coding and temporal coding. Okay. Now that might sound like a lot of information, or might even sound kind of complex and difficult, but I just want to remind you that part of the deal was that this is going to be somewhat complicated of concepts, but that it will be easier to understand when we go through it in class if you've had the chance to kind of work with it before you come to class, because it'll help to adjust some of the vocabulary rockiness, because you'll have already kind of picked up, maybe not the conceptual understanding of what it means, but you'll have picked up a little bit about the concepts, and then, or excuse me, the vocabulary at least, and then I can fill in the gaps on the concepts. Sorry, I'm just practicing using the microphone. It just sounds cooler. I don't have to yell. I can talk in my palate voice. Nice and easy. Make sure your seatbelts are locked and the tray tables are up. We'll be coming in for landing in just a minute. Okay. Anyway, so let's see here. Okay, so how do we, that's kind of how we decide, or not, that's a wrong word. That's kind of how we start to figure out what the sort of tone is of what we're hearing. If you have a tiger growl at you, or Juno's in here and she barks at you because she wants you to pet her, and she's trying to let you know that, hey, this belly ain't going to scratch itself, guys. Right? By the way, she'll probably be coming back in a week or two. You guys want to hang out during class? Okay, I'll bring her in. She'll be pleased. I might even, maybe I'll bring a bag of like some bacon or something. You guys can
Bring
Mm-hmm.
her
really
in for the exams.
bring her in for the exams.
Yeah.
Be a therapy dog. Yeah, they do that now, don't they? During like finals weeks and stuff, they'll have little like puppy sessions, like where you can go and like hang out with a puppy. You guys got it good, man. We didn't have that. We just had beer. Like we just had to drown our sorrows after the fact. We couldn't fix them ahead of time with a beautiful puppy.
You do both.
Yeah, you do both. Okay. Okay, cool. So, so let's say that Juno's in here and she's making some noises. How can you tell where it's coming from? How do you know? How does your brain make sense of location, spatial information?
Oh, because sound's going to hit each ear from a different, slightly different direction.
Yes, you've got kind of an issue, almost like a sound shadow, right? And we'll look at a figure out that in a minute. So the sound waves, it's a lot easier to think of, particularly if like you're talking to me this way, right? So the sound is going to hit this ear before it has a chance to hit the wall and bounce back and hit this ear. We'll see some pictures of that. So that's one way to do it, right? You can compare that entry-level timing, but the question then becomes, that's the difference between the timing when it hits these big floppy skin flaps, right? But how does the brain then make sense of that, right? That's what we're going to talk about with these, particularly these latency differences. So latency means time. Okay? So how many of you guys have ever been late to anything, right? Or you get tired, or you get frustrated when somebody else is late for you? Late. Late. That's about time. Latency and time. Okay? You can also have differences in how intense information is, and we'll look at that too. That helps. These are going to combine to give our brain the information it needs to somehow, dare I say magically, but wonderfully, somehow make sense of these different pieces of information. Okay? Okay. So we have this way that this can happen is basically what we call onset disparity as well as phase disparity. So we'll look at some of that here in a second. So this is that idea of a sound shadow, and Sean, that's what you were saying earlier, right? Okay, so this is kind of the idea that he had. So if you look at this, what we have is, so right, you have a head. That's kind of my thing here, right? Lined up here. And let's say you have sound coming from this speaker. So the sound's moving in this way. It's right. We know that it's a physical stimulus, right? We talked about this compression of air molecules. So that physical stimulus, it has to physically move and travel, right? Now, it's almost identical, but it's microscopically different in terms of its speed, because naturally, anytime, if there's a race, are you going to get to the closer thing first or the further thing first? Naturally, the closer thing, even if it's microscopically different, and that's what this is, microscopic. So this information is going to hit here, and then just a little bit longer, it comes to this layer. So we're talking like really, truly impressive precision. I mean, it's amazing what our biology has adapted to be able to perceive here, right? So this is kind of your sound shadow, because here is kind of that shadow. Just like if this is a light, right, this would be darkness here because it'd be blocked out by a shadow. Right? Kind of like the groundhog here in our month of February. You guys know the groundhog, right? Did he see a shadow this year? I didn't pay attention.
Didn't he like die or something?
No.
I thought I saw something about that like on their Facebook that like the groundhog died like a month early. And it was supposed to.
Oh, no.
So I didn't hear any news on it.
We know they say what's it, if the groundhog sees a shadow, so it's 6 more weeks of winter or something?
Yeah.
You guys ever seen that movie, Groundhog's Day?
It's a great movie.
It's a terrific movie. You guys know who Bill Murray is? Yeah, so he did this great movie in the early 90s called Groundhog's Day, where every single day, so first of all, it's a really interesting movie because it's about having a bad attitude versus a good attitude. And so he's a really snarky, sarcastic, big, fat jerk. He's a weatherman, so he thinks he's a celebrity, but no weathermen are really celebrities, right? So he's like one of those like low-level celebrities, jerks. And he's mean to everybody. He's stuck in this town that he hates. And every day is the same day over and over again. He can't get out of it. Every single thing happens the same. It's like, any of you guys work in a restaurant? Ever? I used to work in a restaurant. We used to run a nightclub on the Jersey Shore. It was just like tourists every single day, and we would say it was Groundhog's Day because it's like the same thing over and over, right? And it kind of messes with you a little bit. You just kind of feel a little insane at times, and because you just want some variety, and that's the movie. But what he finds out is first he takes advantage of everybody because he thinks he's God. He's like, well, the day keeps changing, or nothing changes. I just keep reliving this over and over, so I might as well take advantage of people because he was a big fat jerk. But the way that he ends up beating the game and getting a new day to come is he realizes that maybe I should stop being a jerk, and what if I approached every single day with the attitude of, what can I do for you today? And how can I be a better person? And he spends every waking second of that day, because he knows everybody so well, because he's done it for like 1,000 years now, right? He knows it so well of everything that's going to happen. He also knows how to make everybody have a better day. And he fixes his attitude, has a better day, and he fixes the game, and he beats it. Anyways, Groundhog's Day. So that's seeing the shadow. Now we have a sound shadow. A nice little diversion from auditory information. We're fresh again to dive back in. Okay. Sometimes people have comments about our little tangents, and they think that they're random, but they're there on purpose because I want to give you a break from the academics so that you can be fresh for every new little section. It's kind of like sports. If you're weight training, you just got doing a bunch of bench presses. You don't just go immediately from bench pressing to power cleans. Right? Get your breath for a second. Maybe get a drink of water. Then you go over to the power clean rack and you start doing something else. So now we're going to start talking about these other timing differences. So we have this shadow. And things are going to get to those different ears at different times. And this is sort of just that relative measurement. So you can see these different frequencies from low to high, and they're going to get to your left ear and right ear, right, in relatively different perceptions. So when you have low frequencies, and that sound shadow is not going to affect it. So what's a frequency, right? So when a wave comes like this, up and down, a high frequency is when something happens very frequently. So that sound shadow is going to be really sensitive to blocking out those higher frequency sounds. Right? Or at least it's going to take longer. You're going to have a bigger discrepancy between left ear and right ear, which is where you're going to see that big difference between red and blue lines. But at really low levels, a low wave, there's not enough of space in that sound shadow for it to really detect much differences between left and right ears. So the sound shadow is a useful way to do some things, but it's not a real full account of a way to do everything in terms of deciphering some of this different auditory information from our environment. Sound good? Okay. Okay, and then this is another way to look at that phase disparity. So if we come back 2 slides, right, we talked about onset disparity and phase disparity, okay? So we're going to look at each of these. So you have an onset, which is when does the red and the blue line start? And we're going to see that one starts a little bit differently than the other in time, okay? But we're also going to look at its phase, which means is it an up part or a down part of the graph, right? Are you guys, we've all been in basically good moods and bad moods, or you ever tell someone they're kind of going through a phase, right? Maybe you have a significant other who goes through a phase, right? So that means you're either up or you're down. We graph that, right? It can be either on the upswing or the downswing, and that's what we're going to look at. Sound good? All right. So here's we're looking at 2 things. This is your onset. One turns on before another turns on, right? Yeah? Phase is here. Okay? So look at this. Where it's at is at a different phase. Okay? You could have at one point, see how this is very low? If you go straight up, this other sound is at a much higher part of the phase. This is at a much lower phase. And if you come down here when the blue is at a low phase, where's the red at that point? Oh, wow, that's a lot higher. So that both of those types of information, onset and phase, are really different kinds of information, and the ears are going to get both of that information and be able to start to decipher those differences. Okay. Let's see here. I don't know. We're not going to talk about the duplex theory too much, so don't worry about the duplex theory. But we are going to talk about this idea of a coincidence detector. All right? So, you guys all know what a coincidence is? A coinky-dink? Right? Just seems that some things just happen to happen together at the same time. Okay, so we're going to talk about that. Okay, and a lot of some of this work is done in an area called the nucleus laminaris in birds. And we'll talk about it, particularly in the midbrain, the medial superior olive of the midbrain. And we're going to see some things that can happen. Okay, so here we have our midbrain. Here's that nucleus laminaris. Okay? Now the question is, how does it detect those different sounds that are hitting the ears at different times. So what happens when a sound hits the ear? Remind me. Go ahead, Jasmine.
Okay, so it gets funneled from the travels down the ear canal. It hits the eardrum, which activates the motion of the ossicles, then goes through the collicula and, wait, no,
Cochlea. Cochlea
Cochlea.
is different than the colliculus.
Yeah.
You're right,
Cochlea.
they do sound pretty close, but yeah, they're different.
Yeah, so then fluid causes that to travel to the nerve and then get processed.
Okay, and then it goes in the brain.
Yeah.
Okay, so that's going to happen, let's say, to each ear. But is each ear going to start that process at the same time? Why not? Yeah, because temporal latency, because of the sound shadow, because of everything we just got done talking about, right? So what we have is a race. And there's two runners in this race. And they're going to compete. All right? Now, the runners in this race are going to start, one at each ear. These little neural signals are going to be the runners. But like you just said, they're not going to be able to start at the same time. Does that sound like a fair race? Guess what? Nobody cares. Nature don't care if it's fair. There's not a whole lot of fair things in nature. Not all races are fair. You just got to suck it up and run. That's what these little neurons are going to do. They're say, all right, one's got a head start. Does the one that starts later complain about it? It just starts running. So that's what's going to happen here. You're going to have basically a left side and a right side. Okay, so this is an action potential signal coming in here in blue from the right, and an active potential, whoop, there's a little action potential coming in here from the left on red. Does everybody feel kind of comfortable with this basic framework? All right, let's extrapolate it a little bit more and let's see what happens. So, what you have are these 5 spots. 1, 2, 3, 4, and 5. So you have 5 spots here. These 5 spots aren't really spots. On the cartoon here, they're just dots, but what they represent are essentially neurons. So these neurons are called coincidence detectors. Does anyone know why? Yes, sir.
Can they detect the 2 signals when they overlap?
Yeah, they detect when things coincide. You're absolutely right. All right, so the scientists are doing you a solid here. They came up with a name that tells you what they do in the name. All right, so if I ask you what do coincidence detectors do, you say they detect when things coincide. Right, now neurons are only capable of detecting signals, right? So they detect when signals coincide. Super easy, right? Okay. So, you have this neuron, and it's going to send its signal to coincident detector number 1. But it's also going to continue to send a signal to coincident detector number 2. And then it's going to send this signal to coincident detector number 3. And then it's going to send its signal to coincident detector number 4. And as you might imagine, what's going to happen next, it will send its signal to coincident detector number 5. Okay, so what? Big deal. Who cares? Well, it's really helpful, it turns out, because what's going to happen is when this arrives at cell 1, what's cell 1 going to do?
Nothing.
Nothing. Exactamundo. Why?
Because there's not another signal to, like in the last one, there's not another signal to
To coincide.
coincide.
Yeah. So, coincident detectors care if things coincide, right? And if one thing gets there and another thing doesn't come and match up, then it don't care, which means it won't fire. The thing that will ring its bell, that will turn it on, is when 2 signals coincide and arrive at exactly the same time. So what we do is we just trace this mapping out. Just like this goes to each of our 4 to 5 detectors, this signal coming in from the right also goes to these 4 coincident detectors. So what you can do is you can start to map out the speed by which this happens. And when you do, you can see, so let's say that by the time this signal is coming here on the right ear, so let's say we come back a couple slides. Which ear gets hit first? Remember that. Which ear gets hit second? Remember that too. Right? Remember, big picture, this book, generally, there's always exceptions to rules, right? But generally, the authors are trying to help you out with illustrations that make sense using color. So these colors are red and blue, right? What color is the first one? Where the first stop the sound hits, so it's red, right? Left? Okay. Now we see that mapped out here. Same information. So this is not like a totally new figure. It's representing that previous one. And we see that here too. Also not a totally new figure. It's representing that initial sound shadow figure. Now, this is the same information still. Okay? And if it's easier, I'll even help you out here. Check this out. We will do that. And then we also have, by the way, you can do this too because you own all these slides and these big figures. You should make your own PowerPoints and pull this together to make more sense with your study groups as you each assign each other different sections of a chapter to come to a meeting with. You guys are doing that, right? Okay. Little tip there, word to the wise. It's a really good way to do it. And then okay, so here we are again. So it hit the left ear first, right? Which is red. So it hit here first. And it's zooming. It's race. Off to the races. Dun, 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 dun. It's like a horse race or a greyhound race. Let's stick with the Juno example. Let's get these greyhound doggies racing around chasing this rabbit. Okay? And so by the time the second dog is released, the first dog is already halfway around the track. Right? Okay. So we're trying to find out when these two signals from the left and the right, are going to coincide at the same neuron to send that signal into the brain. So that we can figure out where is the sound coming from in space. And we see, if we trace that out, that by the time that this is sent, right, it's going to come to all these. And, right, so by the way, this is here now. So it still has to travel this distance, right? So it ends up coinciding in this particular example at neuron 5 because it's the closest one to that ear, blue on the right. And in just that little bit of time difference, to go from here to there, it's allowed that signal to travel all the way across that nucleus laminaris. Now they both arrive at the same time here. They make a match made in heaven. They fall in love. They send the signal upwards. The stork comes. It's great. That is a timing difference. You might think of it as an interaural timing difference. Aural means here. Inter. Okay? International means across nations. Interaural, across ears. Timing difference. How do you guys feel about that?
So what happens once they coincide?
Once they coincide, then this neuron will send that signal upwards. Just like we had a tonotopic map that told us the frequency, we have this spatial map of where these different neurons are, and different neurons are going to maintain and preserve this information of sound location, just like other neurons maintain and preserve that information in tonotopic frequency. Yes, Chance?
And those coincidence detectors, those are in our brains or only birds' brains?
We have them too. Okay. They're easily studied in the birds' brains. Yes.
What's this? What does it do for us though?
It allows you to see who's like talking shit behind your back from the left or the right.
So it's like pinpointing locations of sounds?
Yes, it's sound localization is what they call it. Yeah. So there's a way to do localizing sounds, which is really important evolutionarily, because if you're just, hanging at the club and somebody's about to roll up on you, right? And you can hear their chains clanging. So what? Okay. You're going to need to know where that's coming from, right? You're going to know to defend myself here or here, right? You got to hear that. Is the tiger sneaking up on you in the jungle from the left or the right?
So if we didn't have coincidence detectors, would we be getting two separate signals instead of one?
Well, you're already getting two separate signals.
But so you said when they like meet up, then they send the one signal.
Yeah.
So if they didn't like meet up, they would just like eventually just send two separate. at different
Yeah,
times?
you would hear them,
Yeah.
but you wouldn't be able to localize where the sound is coming from. Yeah, I mean, like, I don't know, it's important. You gotta hear, like, so when you're driving on the road, somebody's honking at you because you're about to turn into their lane and they're in your blind spot and you're about to hit them. Doesn't it help, don't you have this like unconscious ability to hear that horn honk and know if it's coming from the right or the left? Wouldn't it suck if you didn't have that? Yeah, you'd have a lot of accidents. You'd be hitting into people. Then they'd be coming after you with their chains in the club. Or just on the side of the street. They'd be like, yo, what's up? You just drove into me. You're like, oh, I'm sorry. They're like, didn't you hear me honk? You're like, yeah, I heard you. They're like, well, then why'd you drive into me? They're like, oh, well, I couldn't figure out if it was coming from the left and the right, so I just decided to keep turning right. There'd be a lot of that. It would be no fun. Yeah. Okay. So that's kind of the idea there. Also, did we talk about the dorsal and ventral streams in the visual field? Same idea is true in auditory stuff. So we have these two main pathways, dorsal and ventral. So dorsal means what kind of direction towards the brain? What you got, Myra? Yeah, more towards the top by your dorsal fin, right? If you're a shark. You guys know who Jimmy Buffett is? No? He's a musician. You guys know the song Margaritaville? Well, he sings it. Your parents probably know it. He's kind of their generation. He sings a lot of songs about getting drunk on a beach in like a tropical island. It's a lot of fun. You should go to his concerts. They're good. They're fun. And anyways, so he sings a lot of these music, and one of his songs is about, the name is Fins. It's about sharks. And it's a metaphor. He talks about this girl who's a bartender down in the islands. She came down from Cincinnati, and basically saying that she's got fins to the left, fins to the right. She's the only girl in town. What it means is she's got sharks circling her at the bar. All of these guys want a piece of that action. They're all hitting on her at the bar, right? And she's just trying to make a buck at work, you know? Just trying to sling some drinks. So he's saying you got all these sharks. So it's a popular drinking song at these concerts, and they all say, and at the time they have like a whole like group sort of. It's like you remember last class we talked about You down with OPP? Yeah, you know me. It's like a white baby boomer version of that. They say fins to the left, fins to the right, and they all put fins on their head. So this is the dorsal fin. So the dorsal stream is going to be closer to the top of the brain where that dorsal sort of activity is, by that dorsal fin. Okay? So you can remember that with margaritas at these concerts, right? You got the fins, the dorsal fins, and the dorsal pathway. So ventral, of course, you just think of as the opposite, right? That means more towards the bottom. So you're going to have these dorsal and ventral pathways. So the dorsal stream is going to include basically the parietal lobe, and the ventral stream is going to go down to the temporal lobe. Now this is true for both vision and audition. That is, seeing and hearing. And the dorsal pathway tends to involve a lot more spatial information. Like, where is this information happening? The ventral information is more specifically about details of like what the sound is. That is, its components. And that's true for vision too. So a lot of times you'll see a dorsal and ventral pathways mean the where and the what pathway. Because the pathway tends to, generically speaking, carry information about what it is that's happening in the ventral stream into the temporal lobe, or carry information about what is happening in the dorsal stream to the parietal lobe. Guys feel pretty good about that? Okay. Let's skip through some of these. We're not going to talk about amusia or any of the clinical stuff right now. Okay. I'm not going to talk about the implants and deafness right now in class, but what we got on time? Okay. How many of you guys want extra credit on the test? Are you sure? Okay. Maybe I'll put questions on there about deafness and that section that we did not cover. And if you get that, maybe that'll be extra credit. Sensory neural deafness and things like that. Like I said, maybe. I haven't made the test yet. We'll see how it shapes up. But otherwise, the translation is that you don't need to know that for the test. Does that sound fair? Okay, cool. All right. Who can tell us anything about the vestibular system? Yeah, Rose.
General idea of
Yeah.
what it is? Okay, so it's like how our body tells where it's at in space, and then in the semicircular canals, the fluid filled, and there's 3 of them, so depending on which way it's pitch, yaw, and roll.
Yeah.
Pitch, yaw, and roll, and like basically wherever the fluid moves, that's how your body, or that's how your brain is able to tell where exactly the body is in space, and there's like other stuff that goes along.
Great job, yeah. That's a lot of information there. We're going to break that down, yeah. So basically the vestibular system that we will talk about is the system that we have for kind of telling us where we are and sort of our position in space. Whether we're on a merry-go-round or a roller coaster or a fighter jet, we'll have those sort of 3 dimensions of direction, which is pitch, roll, and yaw. And we'll talk about that in a little bit more detail. And it's also our system that'll tell us about movement. That's just straight, or movement that when we stop movement. So we'll call it linear acceleration. How many of you guys put the pedal to the metal when you speed off from a stoplight and you got to get away from the fuzz? Yeah, you peel out, right? What happens to your head? Comes back. right? Based upon that acceleration in a linear direction forward, right? So when you move the car forward, in linear acceleration, you have that response where your head goes like this. It's basically the response to gravity, right? We're going to look at how we have systems in the brain that are designed and optimized to really serve our abilities to detect that. Sound like a good plan? And people guess, it's a big fat guess, that it might be related to motion sickness. The verdict is still out, because we haven't solved that conclusively. But we'll get to that too. Okay, so let's talk about some things that Rosie mentioned. Pitch, yaw, roll, and yaw. So these are 3 directions of movement. And you can talk about them and I'll talk about them a lot in terms of an airplane, because I'm a pilot and that's how I think about it, and we use it to describe a lot of things in aviation. But we'll try to connect it to other things too. So we have your airplane, right? You have your wings. Pitch means nodding your head up and down. The nose of the airplane coming up and down. Okay? So pitch is like a baseball pitcher. That direction of the arm. This is pitch. That nose of the airplane. You're either going to put the airplane nose up or the airplane nose down. So these are my wings, either coming up or down. Pitch. Now, if these are my wings, how would I roll? Yeah, barrel roll. Yeah, no, yeah, I like it. All right, yeah. So you do some aerobatics and you do a barrel roll this way or this way, right? Like Tupac said, picture me rolling. Yeah, I ain't mad at you. Okay. You guys know Tupac? Okay, so just picture me rolling. So you're going to roll left or roll right. You're a barrel roll in a dogfight in the air, right? Like in that movie Top Gun. You guys know Top Gun, that volleyball movie with the pilots? Yeah. You guys haven't seen Top Gun, you don't know why that's funny. But how many of you guys have not seen Top Gun?
Oh my gosh.
It's okay. It's a really good movie. Anyways. So it's a movie about fighter pilots in the Navy, actually down here in Southern California. And so you have dogfights in the air. They could be roll left, roll right, right? Okay. Now the other type of movement, and this is actually from the movie, there was an accident in the air. You guys remember when Goose, I guess it's from 1986, so I can say Goose died, right? Not like too much of a spoiler, it's from 1986. Okay, so Goose dies. He's one of the characters. And when they have the accident board, they have to investigate. They say, why did he die? Was it the pilot's fault? Was it the airplane's fault? Et cetera, et cetera. And so the Navy's investigating, and they say that he had an excess yaw rate, is what they say. And generally, nobody pays attention to that in the movie because they're still thinking about the volleyball scene. And so, it's when all the pilots are playing volleyball with their shirts off, they're all oiled up and whatnot. So people like that scene. Anyways, so they say excess yaw rate. So it was a flat spin out at sea, and then Goose crashed because he couldn't get out of the flat spin. What's a flat spin mean if you're in an airplane? I'm spinning. Yeah, this is yaw. This is our yaw direction. Okay? So yaw, roll, and pitch. Okay? Okay, so we're going to have some stuff. Let me see if I can show some things. Okay, so here we have pitch, roll, and yaw. Okay, so here's pitch. Right? Roll is over here, right? And yaw is this direction. Okay? And you'll have X, Y, and Z. So you guys know a Cartesian coordinate, right? X and Y, like in geometry. You guys know why they call it Cartesian? A guy named Descartes invented it. You know, the guy who talked about the mind-body problem, and the pineal gland being the seat of the soul, and then also played his hand in philosophy and said, I think, therefore I am? Yeah, he made some pretty big contributions to geometry, too. Isn't that wild? It's the Cartesian plane, x and y coordinate. That's our x, that's our y, and z goes in the other direction. So we'll have that, and we'll see these things are called semicircular canals, and there's 3 of them. Okay? So you have 1, 2, and 3. These 3 semicircular canals are going to allow us to detect our 3-dimensional movements. That is pitch, roll, and yaw. And they're fluid-filled canals. Just like we talked about the cochlea having the canal in the cochlea. These are canals that have fluid in them in a similar way. And that fluid is going to move hair cells. Just like we talked about hair cells being moved in the auditory system. So, this fluid can move in these 3 different ways. See how they're oriented? This way, and then this way? So when you have movement in those 3 different directions, that fluid is going to move, and it's going to bend those hair cells. Those hair cells live here, here, and here. So if you have a hair cell that lives here, and you got to get it to move, you need this fluid to move in this canal in this direction. And then it'll bump this hair cell, it'll move it, it'll send a neural signal into the brain and tell you, hey, it's moving in the X direction. Probably nose down or nose high. Change the airplane a little bit so you don't crash. And vice versa for that and that direction. So let's take a closer look at some of the things that we have, some of the vocabulary, okay? Okay, so our vestibular system, we talked about these semicircular canals now. They're fluid-filled tubes, and they're in those 3 different places. We also have things called utricles and saccules. Okay? Now here's like reason number a gajillion why you're going to fight a losing battle if you just try to rote memorize everything for this class. Because this stuff gets complicated. You got to know what it is, rather than just regurgitate it back. Right? These are funky words. How many of you guys just hang out with your friends and like shoot the shit and talk about utricles and saccules? Yeah, I mean, Kanye's not spitting about it in any rap songs. You know, like it's not, there's no like pop cultural references to it. You're never gonna see this stuff unless you've already studied it for hours and you've already read it several times and drew it in your coloring books and traced it and made slides about it to show to your study groups, right? If you haven't done that, that's okay, but you want to start doing that. That's how you're going to practice using these terms. So you got the utricle and the saccule. The utricle is going to basically have fluid, and it's going to respond to these static head positions. And the saccule is 2. We'll take a look at that too. Okay? So they're going to be different head positions that they're going to respond to. Now let's dial into this a little bit more in depth. And let's look at this thing called an ampulla. Okay? So the utricle and the saccule, let's come back over here. Okay? So here's our semicircular canals. We've already talked about those, right? We've got that we'll detect the X position, the Y position, and the Z position, and the, X, Y, and Z. Pitch, roll, and yaw. These are your ampulla within those semicircular canals, or at the base of them. You also have these things called a utricle and a saccule. Okay? So these are what's going to help detect sort of linear accelerations. So when we talked about hitting the gas pedal down at a red light and speeding off to get away from the popo, right? Get the 5-0 hot on your tail. You're like, man, I got to get the heck up on out of here. So you slam the pedal and you get out of there. Your head comes back as you're speeding down the runway, blasting off to Palm Springs. Right? Okay, so when you're doing that, are you rotating in X, Y, and Z? No. Okay. Instead, you're just going straight in a linear way, in a line. That's where these utricle and saccules are going to come in. Linear movement. Okay? Now, and I guess here's the other thing, right? And you can see, this is our cochlea. We've already studied the cochlea. We already know about that, so we're going to ignore this. You can see, though, that anatomically, this vestibular system is right next to the auditory system, and they're both going to send information out through these yellow nerves, right? It's going to form the vestibulocochlear nerve, one of those cranial nerves. I think it's the 8th. I might be wrong. But I think it's the 8th. So that's what these yellow lines are. Now, let's come back again here. Okay, so when we talk about that linear acceleration, okay, that linear movement is going to be detected by these things called otoliths. And I think, if my memory serves me right, it was last class, I think it was Rosie, I think you asked in the beginning of class for me to distinguish between
Monoliths and
ossicles and otoliths. Somebody, maybe.
I was just wondering what the difference between the hair cells was.
Ah, okay.
But
Well,
yeah, but I know somebody brought up.
yeah, somebody talked about ossicles and otoliths. So here, we talked a lot about ossicles last time, right? The incus, the malleus, and the stapes. The hammer, the anvil, and the stirrup. Okay. So, now we're talking about otoliths. I'm going to look at some pictures in a little bit. But let's get the idea down, okay? So, this is definitely, it's a little new agey. Okay? Because there's these crystals. Okay? So many of you guys are into like new age stuff. Now we're going to start talking about these little crystals. Now these crystals hang out in this gelatinous mass. That's a really dorky way of saying jello. How many of you guys ever make jello? Alright? It's kind of delicious. You guys ever make a fruitcake for Christmas? You guys ever seen one? Right, you get a bunch of stuff baked into jello. You guys watch The Office? How many of you guys like the prank that Jim pulled on Dwight where he put the stapler in a jello mold? That was kind of good. Okay, so he had a lot of good pranks on Dwight, didn't he? Yeah. So you got jello, and jello kind of shakes around a little bit. It's like blah, 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 right? Okay. So let's say you've got that fruit in the jello. Now let's say particularly you put like stuff on top of it, right? And let's say it's heavy on the top of that, bunch of strawberries on top of that jello mold, like kind of baked into that top layer, right on top, like with frosting and stuff, right? Now let's say you're kind of walking up the stairs to a friend's house to deliver this jello mold that you made because you're just a nice friend, right? And you start to stumble over the stairs, and all the weight of the strawberry starts to kind of pull it a little bit, right? You guys can imagine, right, that the strawberries get to move because the jello is kind of flexible. That's what's happening here. So there's this gelatinous membrane that is this jello. And the otoliths are these crystals that sit there. And those crystals are kind of like little rocks, like little baby rocks. And because they have mass, they're sensitive to gravity. And just like when you start to stumble with that jello mold over the stairs or something, and the strawberries on top have mass and they start to pull, that's what's going to happen here. You're going to have that gelatinous mass with the gelatinous sort of tissue, and the mass, the weight of these crystals on top of it is going to pull it just a little bit. And we'll see some examples of that. And that's how we're going to be sensitive to linear motion. Okay? So, let's take a look here. We already looked at these semicircular canals. Now, this is within the semicircular canal on top. So let's come back here and just take a quick, well, this is easy. Let's just look at these. These are semicircular canals, right? Now remember we said at the base of these ampullae? Okay, there's 3 ampullae. This one, and this one. Let's just focus on one of them, right? So whatever, just pick your favorite one. Okay, so let's just say green is our favorite one. Now inside there, we're gonna look. See how there's this little peak of stuff? We're gonna zoom in on that in the next slide. And we'll find out that there's more of this sort of fluid and sort of gelatinous mass, and kind of baked within it are some hair cells. And they're gonna bend different ways, just like we saw before. Okay, so on top here is what I want to focus on. Okay, because the top is what's going to be the semicircular canal, ampullae, and the top is what's going to give us that rotational movement direction. That is XYZ, pitch, roll, yaw. Okay, so here we have the whole ampulla. Okay, within it is this thing called the cupula, little cup, right? And within the cupula are these hair cells. See those little hair cells within there? They look like little seagrass wisping around. Okay, so as this fluid goes through this canal, now this is different, ignore that one, right? It goes through here and then it'll loop back around, right? That fluid is going to bend it just like this. You see how they're bent? That's what's going to send that action potential down through these neurons. These neurons are then going to send their afferent signals into the brain through the vestibular cochlear nerve that is cranial nerve 8. See how that works? So it's a mechanical transduction process, right? So mechanically you have fluid moving, shifting that cupula within the ampulla in which the hair cells are living and hanging out. And that's going to move those hair cells, it's going to bend them so that it opens those channels, allowing them to be depolarized and send those action potentials out. How do you feel about that? Now here's the thing. As that fluid is coming this way, let's say you're just flowing around in a merry-go-round. They adapt. You just get used to moving in that direction, don't you? You guys remember being on a merry-go-round? Okay. And you're fine, right? You might even be able to talk to people, yell, faster, faster. Where's the problem come in with merry-go-rounds? Yeah, when you stop, right? All right, you got a kid, right?
Yeah.
How old?
10 months.
Okay, so they're not really the age yet for merry-go-rounds at 10 months, right? You don't really want to spin them around like crazy, but at least not yet. And so the problem is when it stops, because in the meantime, it's adapted. But once it stops, then all the fluids has to reset the other way. And that's when you get all stumbling around a little bit, right? Everything's kind of readjusting. Because now you get those kind of mismatched signals. Okay? So, anybody else ever fly airplanes before? I'll give you an example. So, this is what we did. I actually went to the FAA this summer in Oklahoma City, which is where their national headquarters are for operations in aeromedical institutes. And they study illusions that pilots have based on those hair cells that can kill you if the pilots don't do it right. When they fly your airplanes, you get an illusion. It can get just like you think you get off the merry-go-round and you think you're moving one way, right? So you walk this other way instead and everybody laughs at you. Right? So if a pilot thinks they're going one way, but they're actually going the other, and they turn the airplane in the opposite direction, boom! No bueno. Big accident, right? So we really care about these illusions that can take place because of misplaced hair cells in your semicircular canals. Yeah.
So is this connected to, when surfers, what's called burst their eardrum? Because after they get like rolled in a wave and they swim the wrong way, and they go too deep and their eardrum bursts. Is that like the same, is that related to the vestibular system?
It can be. I think that Yeah, it's a temperature issue, and I don't remember the detail of it. I'll have to look it up. It's a good question, Chaz. Let me see. I think I have an example of something here. Hold on here. Let's see. Oh yeah. So this is the Coriolis illusion. Let's see if we have here. So what you do is you take a pilot, or anybody, it doesn't have to be a pilot, has nothing to do with being a pilot actually at all. You put them in this chair, just kind of like this, and you cover up their other inputs, auditory and visual. You go, you spin around like a merry-go-round, but you ask them to give you a signal, to tell you what direction do they think they're moving in. And they use their thumbs. Am I going this way, or am I just staying stationary? Okay, so I haven't looked at all of these. We'll look at them. Some are probably crappy, some are probably better. But let's see if we have a couple. So I got to do this summer at the FAA. It was really, really interesting because it scares the crap out of you as a pilot because you realize how completely vulnerable we are to these illusions. So let's take a look. So see how he had to kind of adjust? That was that Coriolis illusion. Okay, let's see here. Hold on here.
Like I said, maybe you guys will be quiet. Just watch his thumb. And the other thing is, I'm not spinning him real fast.
Look at his thumbs. Starting to get more straight up, even though he's still moving. See? It's desensitizing. He thinks he's fine. Even though he could be in a total diving turn. Alright, let's see if we what else we got here. Let's see here. We got a couple different ones.
Hi, and welcome.
Let's see. Let's do it's the Coriolis illusion. Maybe.
So you're gonna start feeling that right-hand turn. You feel the machine start to move.
So now it's going to be spinning as well. I got to do this too. This flight simulator. It's pretty cool.
So the flying is really more distraction when they're trying to keep you busy. You go for that's fine, if you don't, that's fine. Don't really much you can still go
So the pilot in there with a
Once again,
full
control
cockpit
pitch and
display.
roll, we have control of y'all. We get this machine to work properly. Our first vestibular illusion is going to be the Coriolis illusion. Just time to really reach down, touch that pencil, look all the way down, touch the pencil. Quickly bring your head up.
There's a video of
Now look up towards the ceiling. You're looking up towards the ceiling. Look over your right shoulder. Look over your left shoulder. Back to your instruments. That is that humming sensation. That is the Coriolis illusion. Get rid of that illusion. Focus just on one instrument and keep your head still.
Can't really see it real clear right now.
So right now the machine is talking about the Coriolis illusion, what to expect. down in the position and move it as slow as possible. We'll highly suggest anytime you feel that humming sensation where you think you might make it a little slow, have no reason to look down the floor.
Okay, so let's look at a couple other ones. Maybe this one too. There you go.
Right, okay.
This is winds, actually.
To witness the effect of this rotation on a traveling object, he has set up a revolving camera.
This is actually for a different Coriolis effect on winds, so we'll ignore that for a second. So anyways, let's look at this other thing that we were talking about, this linear acceleration. Oh, yeah.
Would the Coriolis effect only be for the yaw, or would it be for the roll too?
Both. Different directions, depending on where you're going. Yeah. So, we focus now on these otoliths. So now we're talking about linear acceleration, right? So you press the gas pedal, go really fast, your head comes back, right? So now your head comes back, you're looking up. So this is normal, hanging out. You slam on the gas. Zooming ahead, your head goes back. See how that forces this to come up? And the gravity of that, the weight of this pulls this down. Which you have these little links of these hair cells, penicilia, right? Then the weight of these crystals are going to pull that jello just enough to allow these tip links to open, to depolarize and send a neural signal. See the usefulness there, that jello and those crystal masses? Okay. Now, what happens if the light turns red right in front of you? Slam on the brakes, right? Where's your head go? See how this slides forward? It's also a way to detect linear acceleration. But in this case, it's a fast decrease of linear acceleration. It's linear deceleration. Right? Now this is talking about just in a linear way, in a line. So that's how this is going to happen. And now they're going to walk this way or this way. Does that make sense? Now these are going to happen in the saccule and the utricle. Okay. Let's look at some other illusions that you can give. So that can be a real problem as a pilot, because let's say that you don't have any visual information coming in, which, by the way, these information from the visual system, as it gets sent out through the vestibular cochlear nerve into the brain, it gets tightly coupled with our visual information. So there's things called like a vestibular ocular reflex. That is, your eyes can stay fixated on something while you move around, right? Because there's a tight coupling of these neural signals between the visual and the vestibular systems. Now, let's think about that. What happens when you take one of those away? Well, when we trained as pilots, this actually happened to me. I took off, where were we? Somewhere by Palomar or Torrance. You guys know where Torrance is? Right by LA? Yeah, sort of right by Los Angeles, right? It was really cloudy one night. So we landed there. It was my first and only time I've ever been there. I don't know the area. Oh, here it is. Anyways, take off, but it's really cloudy, so I don't have any visual information as a pilot. So you're taking off. Now, as you take off, you push full power in. So you go really fast. It's like pushing the gas pedal down at the green light. So as I'm accelerating in the airplane, and I'm starting to climb a little bit, I'm going really fast, but I don't have any visual information to give me my field of reference. So I don't actually know where I am. I'm in the clouds, and you have to fly just by the instruments of your dials. But I'm going really fast, right? What's happening when I'm going really fast? Right? I'm getting this sense that my head is coming upwards like this, even though my head has not actually gone up in that regard. But the force of gravity has sent this backwards, so now My head thinks it's up like this. So if my head thinks I'm up like this, what do I want to do with the airplane? So if I'm not very high and I'm just taking off, and I do this with the airplane, what's going to happen? You have an illusion that can be deadly. Pretty cool, huh? I mean, that we know about it, that we figured it out, it's not cool that it happens. So, anyways, I figured I'd show you guys. Do you guys want to see some videos of some of these illusions? See it like in an applicable way of how they train pilots? Okay. Let's see here. So we got a couple different ones here. Some are longer than others. Some are shorter. Let's start with the short ones. And because spatial disorientation, as you imagine, spatial disorientation is really devastating in flight. Let's see this one here. We're just going to kind of see how they go.
The moment an aircraft flies into the clouds, pilots no longer have any outside visual references that can give them a sense of direction. Because of that, they may have the feeling that the aircraft is flying horizontally, while in reality it is banked and making a turn. This feeling is called spatial disorientation, and it can be a contributory factor in serious aircraft accidents. Spatial disorientation is an overpowering feeling, one that a pilot can only overcome by systematically checking the flight instruments.
Because we can't trust our brain.
But if a pilot is distracted from the
tricks on us,
instruments
these
and
illusions.
he guides the aircraft by his senses, sensory illusions possibly form a serious threat. That's because in flight, the organs of balance give the pilot misleading information
There's the
about the
vestibular
aircraft
system.
motion and orientation. Boeing and TNO have now developed a tool which can analyze and visualize the dangers of spatial disorientation. SDIT compares the actual movements of the aircraft with the calculated perception by the organ of balance and creates a visual representation of the differences. SDIT enables accident investigators to analyze recordings of the aircraft movements prior to and during aviation accidents in a better way, which will make the tool suitable for helicopter operations in situations with...
That seems like an advertisement for Boeing, which is interesting that they've developed something like that. Like they were talking about that big difference, right, between perception and reality. What we think might be happening might not always be what's really happening. So it's something to keep in mind. So let's take a look at a couple other examples maybe we can dig up here. We already did this one. 3 minutes. Started a 3-minute one.
It has been a smooth flight. The aircraft is on final approach and about to reach Category 1 ILS, decision height of 200 feet in instrument meteorological conditions. The captain is flying. His co-pilot is looking at the instruments and at 300 feet calls 100 above. At 200 feet, the co-pilot calls decide. The captain responds with go around, sets toga thrust and begins to pitch up. Contrary to SOPs, he looks outside but has no visual references and consequently perceives the acceleration as a false pitch attitude. Due to this somatographic illusion, the captain applies stick down, quite the opposite of the movement that's required. As the pitch down continues, the aircraft starts descending instead of climbing. Earlier in the cockpit, the co-pilot stayed on the instruments and saw the pitch attitude reducing, the airspeed increasing, and he called out, pitch. The captain only hesitantly responded, and the aircraft continued to lose height. Almost at the same time, don't sink sounded due to height loss. This supported the captain's acceptance of the co-pilot's call, and collision with the ground was averted. Accident and serious incident investigations have shown that collision with terrain is likely if the pilot flying is affected by sensory illusion and fails to respond to the pilot monitoring alert. But go around.
Here. Did you guys follow that? All right, so it was kind of, so I gave my example about taking off from Torrance. Here's the same thing. Now they're trying to land. And so they're doing a type of landing called a precision landing, which means landing without looking outside because it's complete crazy clouds. You can't see the runway, you can't see anything, so you have to trust the instruments. And it's telling him to look outside, this height where you have to make a decision. Can I see the runway? He can't. So he says, I'm going to go around and try again. So he starts to rise and climb with the airplane. But remember it said he had the visual illusion, where he felt like he was climbing too much, so he started putting the nose down. And so his nose started going down, 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 down. Now we said, remember the earlier video, we said they have to trust the instruments because our vestibular system, our biology, will give us tricks. Our brain will play tricks on us. And thank God he did, especially he had a secondary person there, where the person could say, hey, our instruments are telling us we're actually going down. And he's like, oh, that's weird, I feel like we're going up. So if you're ever in that position and you're a pilot, we're trained excessively to say, you know, whenever there's a discrepancy, who wins?
The machine.
You or the machine? Machine, always. Right? In this case, you should doubt yourself. Because we know they study, in the flight training, they study these vestibular systems to understand these kinds of illusions that we can be subjected to. And so here they're going around. Let's see what else they got next here.
Pilots need to react timely and with precision, toga thrust, adapt or change configuration for the missed approach, and once positive climb is confirmed and called, gear up. Both pilots should fully rely on their flight instruments throughout the approach and go-around. But it's absolutely critical that the pilot monitoring always stays on instruments and does not delay an alert call-out if the pilot flying fails to maintain go-around.
So see there, your sense, you feel like you're coming up here, when in reality you're actually dropping.
Pitch attitude. Disorientation caused by sensory illusion should then be primarily a risk for the pilot flying only.
See how that head moving, acceleration and deceleration, do you see how that fits onto our otolith organs? Yeah.
Facial disorientation can occur as any go-around begins, particularly
Let's
if
see here. Let's see what else we got here. No, it's okay.
That's also why we get motion sickness on like a boat, right?
It can be, yeah. So people talk about particularly motion sickness, let's say in an airplane, right? And again, this is kind of what I alluded to in the beginning of class. This is the idea, the hypothesis we're working on. It's still kind of shaky, actually, in terms of data. But we think that that's one reason why there's air sickness, or car sicknesses, there's a mismatch of our visual and our vestibular system. So usually in real life, you have your visual information tells you the same thing that your vestibular information is telling you. But in this case, if you're in an airplane and you're moving around like crazy, your vestibular system is going to feel it, but your vision just looks normal. Everything in the airplane is just the airplane. You can't see outside, right? So you can't see that the horizon changes or whatever. So all you feel is that you're getting shaken around, but you're looking normal. And they say one idea is that that is basically a parallel. You're tricking your body to think it's poisoned. Why would your body think you're poisoned? Well, when you eat some poison, you get all messed up. It's kind of like drinking a bottle of tequila. Drink a bottle of tequila, the room spins, right? Yeah, so you feel like the room is spinning, even though it's not. There's this mismatch of, in this case, when you're drunk, right, your vision looks like it's spinning, but your vestibular system is feeling what? Nothing, because you're face down on the bathroom floor, hugging the toilet. Right? So you're not physically moving at all. You're like, oh my God, I'm never drinking again. Right? So you have this mismatch of the visual and the vestibular system. Same thing in air sickness, whatever. So your body thinks it's poison. What's a great way to survive? If you think, if you think you ate some bad berries, or if you think that in your kingdom, your servants poisoned your food to steal your crown, what do you got to do? You got to upchuck that sucker fast. So you throw up. That's kind of the idea that people have put forward as to what might underlie that. A little shaky on the evidence still. Any guys ever been seasick before? Yeah. We did, Erica was in Grenada for a couple years, and we would go down there and do like sailing and deep sea fishing, and the waves were just crazy sometimes. And we had a cabin. We actually had the honeymoon suite in this big ship right at the front of it. And it was a really nice little cabin and room. And we'd be moving up and down, and she would get seasick. And The part where it got really bad, where even I started to get woozy, as soon as you go into the little bathroom cabin, and you go below deck, you're in the bathroom, there's no outside little porthole and window. And now you're just in this tiny little room, and now you're getting tossed around like crazy. Again, you've lost this visual input to stabilize the vestibular feeling. Whenever you're out seasick or car sick, what's a good thing to do is you look at the horizon, you can start to match some visual information to kind of quell the vestibular. So let's take a look here. Let's see if there's any other cool things here. I wonder. This guy's just talking, no illustrations. I wonder. That's a little bit more aviation. Let's see here. See if there's a good one. This is from the 90s. How about that?
Humans possess the natural ability to maintain body orientation and posture in relation to their surrounding environment, whether at rest or motion. Good spatial orientation is due to effective perception and interpretation of your senses: visual, vestibular, proprioceptive, and auditory information. Two modules will be used to discuss how your body maintains spatial orientation. This module will focus on the vestibular and proprioceptive systems. The second module will discuss the visual system. Are detected by the vestibular system. Flight requires an understanding of your sensory organs and knowing how you can compensate for their limitations. Visual references provide the dominant sensory information to maintain spatial orientation. This is especially true when your body and/or environment are in motion. Changes in linear, angular, or gravitational acceleration are detected by the vestibular system. Information gathered is mentally compared to the visual information being received. When flying your aircraft in instrument conditions, without outside visual information available, the aircraft flight instruments are the only means to confirm your aircraft position. Under these conditions, vestibular system can give false illusions. The vestibular system is located in the inner ear. This system is the size of a pencil eraser and contains two distinct structures: the semicircular canals and the otolith organs. The semicircular canals detect changes in angular acceleration, while the otolith organs detect changes in linear acceleration. Both of these provide information regarding your body's position and movement relative to Earth.
It's a good soundtrack, huh?
3 half-circular interconnected tubes inside each ear comprise the semicircular canals. The semicircular canals of both inner ears monitor angular acceleration and sense rotation in 3 dimensions. The planes of the 3 canals in each ear correspond to the roll, pitch, or yaw motions of an aircraft. Each canal is filled with a thick fluid and contains hair cells. The hair cells move as the fluid moves inside the canal. The fluid moves in response to angular acceleration. The movement of the hair cells is similar to the movement of seaweed caused by ocean current. If your head is kept still and the airplane is flying straight and level, the fluid in the canals will not move and the hair cells will remain erect. This signals your brain that there is no rotational acceleration and can be confirmed either visually or by instruments. The somatogyral illusions concerns false sensations about the magnitude and/or false perception of rotation in its actual absence.
It's scary, man.
The graveyard
It is
spiral
really
and
scary.
spin due to somatogyral illusions, are two of the oldest in aviation.
I've flown and stuff like this, man. You need
The
to brown shorts with
semicircular
you, man.
canal is
It's-
activated when you accelerate rapidly into a turn, in this case a left turn. The fluid within the semicircular canal lags behind the accelerated canal walls and bends the hair cells to the right, or in the direction opposite that of the imposed acceleration. The brain interprets the movement of the hair cells to the right as angular movement to the left. If the turn continues at a constant rate for several seconds or longer, the motion of fluid catches up with the canal walls. The hair cells are no longer bent, and the brain receives the completely false impression that turning has stopped.
That's when the guy put his thumbs back
level
up.
flight, the fluid inside the canal will continue to move during the rollout, and even for a while after the turn stops. This will send a signal to the brain indicating that you are turning in the opposite direction, an illusion. You can observe this reaction by using a Barony chair to simulate a constant rate turn. Blindfold or place goggles on a pilot to deprive the subject of visual cues and secure them in the Barony chair. The subject is then told to indicate the rotation of turn by using thumb signals. As the rotation begins, the subject is aware of the movement. But will gradually lose the turning sensation as the fluid catches up to the rotation. As the chair is brought to a sudden stop, the canal fluid continues to move for a short period of time, and the hair cells are bent to the left. This gives the subject the illusion of turning opposite the original direction. If this sensory illusion is believed, you may try to counteract it by inappropriately turning the aircraft in the original direction. Now, let's examine some other in-flight illusions that are caused by the semicircular.
All right, who wants to try it? Let's give it a try. We got a chair that rotates. Got some goggles that'll block out your vision. Who wants to go for a ride? Anybody?
I don't know if anybody wants them, there's sound canceling.
Let's just try it without the. Let's see if we can pull it off cheating without the auditory input. No volunteers? All right, let's do it, Jasmine. Okay, so these you cannot see through. You guys see that? All right, so we'll ask her to wear them now. Usually you put the headphones on too. We'll see if we can see the sort of cheat around it. And I'll have you put them on, and we'll kind of move you a couple different ways, and then I'll start spinning you. But remember, you got to tell us, keep your both thumbs up,
Okay.
and you're going to tell us with your thumbs which direction you think you're moving.
Okay.
Okay? She cheated. Oh, okay, okay. I'm going to try it again a little faster. All right, you ready? Thumbs up. Didn't work. Okay. What way are you moving now?
Stop.
Yeah, you are stopped. I probably went too slow. I didn't want to like really wing it on you guys. But we'll try maybe next time with the, we'll do the headphones because I think our time is up now, right? Okay, we'll get it better next time. We'll try it out. Thanks, Jasmine. Yeah.
So, grandma went in, and we were not, so that means that, so back
question.
Wanna finish it?
Thanks, Lisa.
I'm right about one there, I can bring it.
Oh, okay, cool. If you don't mind, that'd be great. Yeah, kind of cool. $600 device.
Alright, so we have grade checks for housing.
Okay.